Welcome, everybody, to Emerge 2050 webinar series. Welcome back. I hope that you all had a good week. I'd like to welcome back everybody to Emerge 2050 HealthTech Talk webinar series, where we continue the conversations we had on stage at Emerge earlier this year. For those of you that missed last week's discussion, from big data to bedside, is AI revolutionizing cancer treatment with Dr. Sadiq Musat and Dr. Joe. Please, you're welcome to go view it on our YouTube channel, and the link is posted in the Emerge 2050 LinkedIn page. It was such an engaging meeting, and I encourage you all to ask questions in the Q&A section today during today's presentation that we'll address after the presentation. In this series, today we'll be delving into the cutting-edge technologies shaping the future of healthcare. Today's discussion will focus on using BioCorteX's CarbonMirror platform to explore the integrated relationships between antibody-drug conjugates, intra-tumour bacteria, and the tumour microenvironment. So I'm so privileged to welcome back Dr. Nick Sharma, CEO of BioCorteX, who joined us in Dubai in May. Dr. Nick Sharma is senior consultant neurologist, and before co-founding BioCorteX, he was a consultant at the National Hospital of Neurology in London and served as principal investigator at UCL. I would also like to invite Dr. Muhannad Alomari, also known as Mo. He's the CTO and co-founder of BioCorteX with a PhD in computer science and mathematics. Mo's journey in tech began early. He built an autonomous car at the age of 21. He's fluent in 2 human languages and 7 program languages. And Mo's accolades included honorable mention at the Distinguished Dissertation in Europe, awarded by the European Conference for AI. Best Natural Language Processing paper awarded at the Association for Computational Linguistics conferences. And now I'm falling over my words. And Best Machine Learning paper award at the European Conference for AI. His career includes significant contributions as an AI researcher at Rolls-Royce, where he rose to the head of AI across Europe and North America. Mo holds 6 US patents in computer programming and has published 28 papers in AI. Outside of his professional endeavors, Mo enjoys building robots and spending time with his children. I don't know, Mo, where you get the time to be playing with robots. But yes, I'd like to open the floor and, big for exciting day to day and exciting to hear everybody. And I'd like to hand over the floor to Dr. Nick Sharma. Welcome.
Okay, thank you ever so much for the introduction. And Mo is, I think, just struggling with his camera a little bit, but should be online very, very shortly. Look, thank you everybody for taking the time to attend. So really excited to continue the conversation with Emerge 2050. So what we're looking at with BioCorteX is what we want to focus on here is actually looking at the interaction between bacteria and ADCs, and antibody drug conjugates. It's something that as a company we are specifically sort of focusing on. Now, the interaction between bacteria and drugs, give me one second. We think is a core component of what actually happens and why an individual actually responds. And one of the areas that we're focusing on, sort of BioCorteX, and our core mission is trying to capture this relationship between bacteria and our health, and trying to answer the fundamental question that, as a clinician, I think is so important, but also is important from a commercial perspective, say from large pharma's point of view as well. And is this medication going to work? And ultimately, what we're trying to do here is we're trying to save time, because time is the one thing that everybody has a limited amount of, and ultimately can't be bought. And so what we're trying to do is work out whether an individual is likely to respond or not. And then if they're not going to respond, they can save that time and can essentially move on to an asset which they are more likely to respond to. From a commercial perspective, we know that drug development has an annual cost of 0.25 trillion with a success rate of about 4%. So very, very poor success rate. And ultimately what we're able to do is actually help sort of de-risk that. Now, why do drugs interact with bacteria. Now, this is something that goes back, a couple of billion years, actually, when actually bacteria bec became symbiotic with some prokaryote cells. So they became mitochondria, and essentially all mitochondria that we all have that are in all cells essentially are former bacteria. Most of their sort of proteins, so over 50% of the proteins that they still produce are actually bacterial in origin. And we know that mitochondria are essential from an oncology perspective, both from the pathogenesis of cancer, but also in actually determining response. So therefore, what we see is that we see we're actually designing sort of medications to interact with bacteria, because ultimately they often interact with mitochondria. So it's something that we think is just hasn't been looked at for a very long time. Now, the way we think about this is we think about this a little bit like one would with a telephone. So we will design an app, say for an Apple phone, and it's designed for that particular hardware. We'll only work on that hardware. But what we're saying is that that's essentially what we're doing with biotech and with pharma. But we're saying that there's an operating system similar to Apple or Android, etc., that sits in the middle, and it's that operating system that determines the compatibility. Now, my parents are terrible sometimes at updating their operating system and their apps stop working, and I get lots of phone calls about that. And it's exactly the same principle. So what we're saying is that we often design drugs based upon the human hardware, but there's an operating system based upon bacteria which varies geographically that actually determines whether somebody's likely to respond or not. Now, the geographical component we think is really important here because the bacteria actually vary depending on individuals' diets. But when you look at the bacteria at a population level, it actually varies quite markedly between countries. So, for example, if you're living in the UK versus living, say, in UAE or in Dubai versus, say, New York, the bacteria that's likely to be present in your gut and even actually in a tumor is actually likely to be very different. And we think that that variation actually causes some of the differences that we see as clinicians when individuals do or don't respond to medications. So what we've done is we're trying to bring in technology that's used in aerospace. So this is sort of speaking to Mo's background. So he was head of AI at Rolls-Royce, the power company. So that's the aerospace company, not the car company. And from an engineering perspective, you think about a company like SpaceX. They did the self-landing rocket on its ninth attempt. Now, the way they were able to do that was actually run emulations or simulations in silico hundreds of thousands of times. So they were able to fail in silico really quickly, so that on that ninth attempt in the real world, they were able to get that self-landing rocket, which was thought to be completely impossible to do. So what we're doing is what we've done is actually try to build a foundational emulator, which we call CarbonMirror, which we'll talk about a bit, which is our platform, which allows us to run these emulations so that we can try to fail as much as we can in silico. Now, the system is far from perfect, and that we're not saying that we can mimic a tumor in silico completely accurately. We certainly can't. And in the same way that we don't fully understand the equations that govern why an engine works, we don't fully understand, obviously, what happens within the tumor microenvironment. But what we're able to do is actually provide more information than what is currently available and able to look at whether those scenarios are likely to happen or not. Now, we think about this in different ways. So we think about there's the oral drug bacteria interaction. So, we take any medication orally, can interact with the bacteria in the gut. But we've got a very specific focus, and what we're going to focus on here is on the bacteria within the tumor and within the tumor microenvironment. Now, there's a long-standing history of bacteria and cancer, and actually goes back, actually hundreds and potentially, depending on how you look at it, thousands of years. Certainly, more recently, it's become very well recognized that bacteria and the microbiome within the tumor, but also within the gut, has a profound effect both on the pathogenesis of cancer, but also on the treatment response. So, in the landmark sort of paper of the hallmarks of cancer, which has obviously been going for a very long time, microbes are actually now added to that of 2022. So this really speaks to the evidence actually behind this approach. Now, lots of this work was actually done on looking at response to checkpoint inhibitors and showing that in in melanoma, you can actually shift the response by about a third by changing the gut sort of microbiome. But what we're focusing on is actually the bacteria within the tumor itself. And this is one of those situations where we've known about this for about 100 years. You know, we've been able to see the bacteria within the tumor cell itself. And if you look at this is from, you know, all the publications at the bottom here, but you can actually see there are bacteria within the tumor cell itself. And if you think about it, the fact that mitochondria are actually within cells, mitochondria actually move between cells within the tumor microenvironment as well. This is actually very, it's not as remarkable as it sounds. What's also becoming very clear is that these bacteria have different roles. We know that if you use antibiotics and wipe out essentially all of the bacteria, patients don't do as well. And that's actually what actually started, well, one of the areas that really pushed the more recent investigations into this area. If you look at what those bacteria do, say in sort of pancreatic cancer, we know that within the tumor cell itself, the bacteria are actually able to metabolize the therapeutic to an inactive form. And that was shown in preclinical models. And then when it was looked at in the patient population, the more of those bacteria that the patient population had, the worse their outcome was, showing that this isn't an epiphenomenon, that this is actually causal in nature. The other thing that I think is actually worth pointing out, and this is something that we alluded to before, is that there are differences within the bacteria. Hey Mo, this is Mo, my fantastic co-founder and CTO at sort of BioCorteX. Mo, do you want to quickly say hello since you've got your camera working? No, we can't hear you still. That's fine. We can't hear Mo. Um, I'm sure we'll get that sorted out. One of the things that's a core principle of what BioCorteX does is we are very keen to be able to look at the geographical differences of response outside of, including, but also outside of the US and the European populations. So one of the challenges is looking at bacteria in different populations. And one of, you know, if we look at, say, cancer incidence, and this is from a great paper in 2022, showing that actually within, you know, the MENA region and beyond, the incidence of cancer is actually much lower. And this has been attributed to diet and potentially other aspects. But I think one of the things that we've been able to show is that actually the intratumoral bacteria is actually varies at a country level as well. And so one of our core hypotheses and one of the things that we're working with sort of partners within the MENA region is actually looking at sort of tumor samples within that population so that we can see whether those differences are actually responsible for some of the differences in response. Now, quickly onto our technology, and then we're really going to focus on the ADC interactions here. So our tech, I'm going to start and say the first thing is we're not doing Gen AI, and this is not because Mo and I don't really love AI. You know, we both got very strong backgrounds in the AI sort of field, but it's looking at the right tool for the right job. And so what we built with this foundational emulator is based upon first principles. And so it actually maps to biology. So we're not looking at making decisions just because the black box said so, and this is, you know, the black box says you should do this, etc. We really believe that the mechanistic approach is really important. And I think as a clinician, you know, I think understanding the mechanism to being able to explain that to the people that are under my care, I think is hugely important. So the first piece of our technology is actually called carbon knowledge. Now, this is a knowledge graph. I won't go into the details of what the difference between a knowledge graph and a database is, but it essentially connects sparse information. It's what sort of Google's sort of search is actually based upon. Now, we believe this is actually the world's largest biology-based knowledge graph. It has over 15 billion connections. There was a press release, if anybody looks at the LinkedIn sort of profiles, there's a press release literally 48 hours ago, joint between Google and BioCorteX explaining our approach to the technology here. And this has over 5 million microbiome samples from around the world that have been reprocessed against our proprietary library. And that allows us to run these in silico scenarios. We've also got over 150 tumor samples from around the world as well, which allows us again to run these in silico trials. So CarbonMirror is our foundational emulator, and I'll go over this in a little bit of detail. What it is it actually builds up based upon pathways into an in silico representation of a bacteria, which then moves into an in silico representation of a tumor. Now, what we're then able to do at an individual level is then have hundreds of individuals and run in silico trials. Now, each level is imperfect. We don't have all of, we don't know all of the pathways required within a tumor microenvironment. But in the same way of, if you look at an ant colony, you don't necessarily need to get all the interactions between the ants correct to be able to predict what the colony is going to do, and that's called emergent behavior. And it's something that's very well grounded from a neuroscience perspective, and it's something we've been doing for many, many years. And so what we're able to do is unlock the value now, without necessarily understanding all of the particular aspects. And again, this is ongoing. So this is something that is present within the ADC sort of trials. So what we need to be able to do is actually do better than what people are doing now. Now, ADCs, antibody drug conjugates. So they work. So ADCs, they are the next big thing in oncology, and as it's that slide when I'll show you when it comes back, when they work, they really do work, but they do frequently fail. And I think one of the challenges of what we're looking at is to see whether the bacteria in the tumor actually contribute to that failure. Now, when you look at the market of ADCs, it is very large. It's projected to be around 24 billion. By 2030. There are hundreds of ADC trials in ongoing at the moment. Okay, so apparently I can share. Okay. Okay, so Mo, can you give me a thumbs up if you can see my screen? No, nobody can see my screen. Okay. Okay, and Mo does not. Okay, right, let's try this again.
Your screen was visible. Your screen was visible, but Mo, there's definitely something wrong with your microphone. Yes.
Okay, so let's try this for a second time. And here we go.
Yes, it's visible. You can just go slideshow. There we go. Perfect.
Okay, back again. Right. So lots of trials ongoing at the moment with ADCs, which is the key point here to make. But if you look at ADCs, they don't. There's a large failure rate. So over 349 have actually entered the clinic or have been tried. Only 11 have actually been approved by the FDA, and this is over a number of years. 190 are currently undergoing active development, but 148 of them were discontinued. So the question is, what determines that success? And our core hypothesis is whether the bacteria within the tumor are actually part of the reason why they fail, and we have that variance in response. So what is an ADC? I'm aware that everybody's backgrounds will be slightly different. So for the oncologists, forgive me for going into the background here. But ADCs are antibody drug conjugates. So they've got three components. They're relatively simple from a schematic perspective anyway. So there's the antibody component to this, and this is what binds to a unique protein expressed by the cancer cell. There is the linker component, and what the linker does is it actually links onto the payload. Right now, what do each of these things do? So the antibody is essentially the hunter part to it. So what it does is it allows precise targeting of proteins expressed by the cancer cell. And then what that allows it to do is it allows then the payload. So the payload is essentially the killer component to it. So that then gets ingested within the cancer cell, delivers a extremely powerful cytotoxic, which then kills the cancer cell. Now, the linker technology as well is a key component to that about whether how that's actually put together. But fundamentally, this is very, very targeted. And so what in principle this allows the ADC to do is to target sort of cancer cells that are expressing a particular protein and then delivering a significant payload to that cancer cell and therefore, improving survival. Now, what we're going to show you is actually different use cases of how bacteria actually interfere with this. And we're going to talk about these at a very high level and show you some of these in silico trials that we've done. As you can imagine, these are commercially sensitive sort of assets. So we haven't, we're not speaking about specific assets. We're going to talk about things at a general level, but the examples that we're going to show you have been ran on very specific assets, some of them in play, some of them sort of not in play. So the first sort of use case is actually showing the interaction between bacteria and the actual payload component to a cell. So this is actually looking at the gut microbiome, because this is an oral sort of medication. Now, this will in a next phase, this then is actually used as a payload component. And so we'll talk about this particular component first. The second, we're going to show you about where the bacteria within the tumor actually interact with the payload within the tumor microenvironment. And then the final use case that we're going to show you is actually where the bacteria itself produces a protein that actually competes with the ADC binding site. One of the key components about the way that we've done this is that for each of these trials, and we're going to show you trials that we've run in silico, is we've used the same inclusion-exclusion criteria, same geographical sort of location as the trial protocol itself. So what we're allowed to do in this context is we're also, we can vary it. So we can see whether, say, these ADCs would work in a particular population. So, for example, in the MENA region, etc. That's well within our capabilities and is part of our commercial offering. So, focusing on the first use case. So, this is actually looking at cytotoxic and small molecule therapies and showing whether they interact with therapeutics. Now, this is work that we presented, and actually James, who is on the call here, won an award at ASCO about 6 weeks ago. And what this was actually looking at 4 assets that have got a significant difference in their 1-year survival between a Japanese population and a US population. And we think that that is the largest drug-bacteria interaction hiding in plain sight. We're not saying that the drug-bacteria interaction explains all of that variance, but we are saying that there's a significant component to it. So what we did here was we looked at 4 assets. These are the little bars here. And the blue bars are the drug bacteria metabolism within the US. And as you can see, it is higher than in the Japanese population, which is pink. Now, what's been shown across these 4 trials is that there's a difference in 1-year survival of 16%. Now, that's externally validated. That's from an external publication. So that's not internal to BioCorteX. Now, what we're able to show is actually this 1-year survival relates to that drug bacteria interaction. Now, just to be clear, we are not saying that all of this 16% is explained by this drug bacteria interaction, but we're saying a significant amount is. Now, How does that compare to pharmacogenomics? Now, we describe ourselves as pharmacogenomics 2.0 or non-human pharmacogenomics. So what we've done is we've looked at one of those particular studies, and some people may be aware of these studies. So looking at the Japan Clinical Oncology Group and the Southwest Oncology Group, and we picked these because these trials are completely mirrored. So they are identical both in design and in inclusion criteria. So we know that there's no differences there. There are 5 times more greater interactions between the bacteria in the US population than in the Japanese population. Now, in an external analysis of this work, it was shown that the human pharmacogenomics don't explain any survival benefit. But what we're able to show is actually the drug bacteria interactions explain a degree of that survival benefit. Now, the degree is a similar magnitude as smoking. So certainly not everything, but certainly a significant amount that is clinically relevant. So what we've shown you there is what's happening in the gut. But what we're trying to do there is now then bring that into what happens if you use one of those cytotoxic as a payload itself on an ADC. So what we're going to do here is actually look at the payloads, the different sort of payloads here. So this is a particular sort of pharmaceutical company. Its phase 3 trial demonstrated a response rate of this ADC of around 35%, so not fantastic at this point. What the analysis has shown is that the numbers needed to interact on their inclusion criteria was 1 in 3. So 1 in 3 patients essentially had an interaction. So let me show you what we did as well. This was in breast cancer. We mirrored their study design. We looked at 100 sort of breast sample tumors, looking at interacting proteins to see whether there was a difference in the payload activation here. And when the study was around, there was no subgrouping essentially based upon intra-tumour bacteria because it isn't a particular component that's looked at present. But what we were able to show is about a third of the patients actually had an interaction. Now, what we then did was then actually go on to validate this in 2 further cohorts using entirely different data sets. Now, we've got a large partnership that we're about to announce with a phenomenal sort of company in the US that has up-to-date sort of data on tumor sequencing with the latest sort of treatment regimes that we will be reproducing this data in. But what this would allow us to have done actually for the study, had we been engaged with this pharmaceutical company, would have actually been able to identify whether these patients would have actually responded. So there were clear, actionable outcomes. So that's looking at actually the payload component to it. But now thinking about the antibody itself, because again, each of these sort of components can have a different example. Now this is there's a number of receptors that we've sort of processed here. This is in a sort of non-small cell lung cancer sort of population. This is a trial that actually failed. And what we were able to show is that there was bacteria within about a third of the patients, again, showing that there was a differential response, or that there was enough intratumoral bacteria producing a protein that was competing for the binding site. Now, in this particular program, the pharma company had a number of different solid tumors that they could go after. What we were able to do was actually look at the bacterial representations in those different tumors to see what the representation was. Now, in non-small cell lung cancer, that was picked, it was about a third, but there are other tumors where this ADC could be used that actually don't have the same representation. So there's clear, actionable outcomes that are potentially useful for the pharmaceutical that we're in discussions with. So it actually could result in resubmission to the FDA, and it could support additional trials in those particular tumors. So what we're showing you here, sort of briefly, is actually showing you the different potential sort of use cases of our technology to look at how these bacteria interact with tumors. Now, the challenge is one of the things that we always get is, look, why do this computationally? Just get the bacteria and just look to see whether they actually interact. And I think one of the challenges here is that it's a maths problem. Yeah, there are real complexities here about trying to do this in the real world without doing this in silico sort of beforehand. So first of all, from a bacterial point of view, the number of bacteria are phenomenal. Depending on how you count bacteria, they're into billions, potentially into the trillions. They're constantly mutating. Just even actually, describing what they're called is very, very challenging. And actually, very few as a percentage, so less than 5%, in fact, 2 or 3% of bacteria are actually culturable. So actually trying to carry out these experiments within a sort of lab environment is actually very, very challenging. But there are other significant components that make the maths even worse, actually. There's an ever expanding list of antibodies to target. And this is super cool. This is exactly why ADCs are so important. So, this is a fantastic figure from a 2023 paper showing that the different antibodies that are in play at the moment. So, if you look at HER2 and, potentially HER3 or TROP2, etc., there's a very long list of potential antibodies. The grey are the active sort of studies, but as you can see, the pink are the ones that have failed, and the green, which is a very small number, of the ones that have actually been approved. Now, this adds, large degree of complexity to the bacterial interaction aspect. Now, you then add on to that the number of payloads that are actually in play as well, because of course you can actually link, different antibodies with different payloads. These are the payloads as of January the 1st, 2024. This is a large number. No doubt in the last 6 months, this number would have increased dramatically. So even the sheer combinations of ADC, well, the actual antibody itself and the actual payload numbers is actually phenomenal. But you then add into it the complexity from a bacterial perspective about the geographical variation. You can see that the numbers involved are phenomenal. And so what our technology allows us to be able to do is to be able to run this in silico. Now, the other thing that is actually really important is we've presented 3 use cases on the 3 examples. This is a schema that the team have put together about other ways that actually bacteria can interact with ADCs. And so it isn't just as simple as the 3 use cases that we have focused on. There are actually bacteria can be involved in sort of cleavage of the linker, which can cause systemic circulation problems. They can look at actually varying sort of receptor expression itself. And so there are a large number of ways that the bacteria can interact with the ADCs. And from a reductionist point of view, it's quite, it would be nice if we could simply reduce this down to one single mechanism. But what we're able to do with our technology is be able to look at a large number of these mechanisms in place simultaneously. And again, this isn't an AI black box approach. This is something that's actually purely mechanistic. So, for example, when we're working with partners, if they aren't concerned about, say, systemic off-target effects because they don't believe that's relevant for their particular program, we can just simply remove that. But however, we're able to work with them on the specific pathways that they are excited about. So we think that there's a real potential here to actually unlock this entire market with ADCs. We think that, there's been a large number of failures today, as there has been across the entire drug development sort of pipeline. But we think that this is something that can be solved. We think it is solvable, but we think this can only really be solved in silico, which is then obviously followed up with sort of targeted sort of validation. But we believe it has to come that way around because the numbers involved are just simply too large to be able to do this in any sort of laboratory sort of setting. So the key take homes here are ADCs are a phenomenally exciting area that at all the conferences, at ASCO, at all of the major conferences, is some amazing work being done on ADCs, both in the antibody components, the payloads. We haven't really touched upon the linker technologies. There are phenomenal companies out there at the moment designing some really clever linker technology to be able to look at sort of payload deployment, etc. Super exciting work. And I think what we would like to do is be able to help those companies essentially make certain it's going to work when they hit the clinic. Because what we're after doing is trying to help those individuals around the world actually have treatment that works for them. We think that what we're able to do is essentially save time, because ultimately we can do this in a serial manner where we have large, expensive trials. But we think that we need to move faster for our patients and the people that are under our care. And what this does is actually provide a competitive advantage for companies that actually work with us. Now, from the point of view of perfection, and I just want to talk very specifically about this, is that we don't, our system is far from perfect. And so what we're not saying is that, the CarbonMirror technology is going to be absolutely perfect. But what it enables us to do is actually give more insight than what's going on at the moment. Because at the moment it's random. And the expression that we often talk about at BioCorteX is that when you're being chased by a bear, you don't need to outrun the bear. You need to outrun the slowest person. And so, it sounds a bit strange, but the point here is that competitive advantage for companies that partner with us is actually significant. We have different offerings that we do. So, we're officially partnered with Bayer, and we can talk about that in the public domain. We've got Alpha, which is where we look at drug-bacteria interactions for oral medications. We can do this for small molecules. We have Phoenix, which is targeting failed trials, and then we have Carcinus, which is the in silico representation, where we look at ADCs. So this comes back to our overall mission of trying to capture the human bacterial sort of relationship to answer that fundamental question that as a clinician and as a human being, essentially, what we're trying to do is we're trying to save people the time to answer that question of like, is this asset going to work? And having been a clinician for a very long time, it's if you've ever sat on the other side of a desk, if you look at response rates, that not knowing is phenomenally important and causes a huge sort of burden on all of our patients. And so what we're hoping to do is to be able to answer that a little bit, not perfectly, but to say whether this drug would actually work and unlock that value for the pharmaceutical companies and the biotech companies. So we'll stop there. Sort of really happy to take questions. And again, apologies about the technical sort of parts to this. Please reach out if you have any questions about this. And we're always happy to look at people's assets to see whether there is a drug-bacteria interaction, because sometimes the knowledge that there isn't is also phenomenally useful. So I'll stop there and happy to take sort of questions. I'm not certain I suspect this may have to be done via text rather than mics, given the challenges that we've had, but let's see.
Can you hear me that better?
We can, and you don't sound like Mickey Mouse, which is a
wonderful.
Excellent. I don't usually sound like Mickey Mouse, just to say thank you very much. First of all, Nick, thank you very much. This has been actually an exciting session. Thank you for sharing all of that. And for the audience, please do share any questions you have on the science, clinical side, technology, etc. in the Q&A section. If you have any questions, I will be keeping an eye on that for any more questions. But to kick things off, I have a couple of questions myself. So just as a start, and looking at the cancer space, so there are now mounting evidence on the role the bacteria plays in the determining basically response or lack of response in cancer patients, such as the hallmarks of cancers. There's been numerous papers, would be actually also excellent to hear more about those. But in your opinion, why do you think there is a slow adoption for, say, understanding the role the bacteria plays in cancer? And so what are the challenges and what can we do about them?
Yeah, look, that's a great question. And just for the audience, I had no sight of that question before Mo asked it. I think there are a number of challenges. I think first of all, the oncology field is phenomenal at adopting new approaches and new techniques, and they are as a community are really pushing forward with this. So A, C, and R. There was a huge amount of work on the bacteria within tumors. So the community, I think, is hugely primed and ready for this. I think the challenges in the past has actually been twofold. One has been access to data. I think accessing data is challenging, but there have been large initiatives internationally across large sort of populations for open sort of data sets. But I think the other challenge there is actually the computational components to be able to examine those numbers. So, for example, we have over 5 million microbiome samples within our engines, and we could have many, many more than that if we wanted to. We're really strict about our criteria, but if you were to use standard state-of-the-art tools to even process those, it would take years. Or even if you did this with a brute force approach with AWS or a cloud provider, it would be in the hundreds of thousands. And I think what we've been able to do by being able to perform that very, very quickly, and actually being able to reprocess data much more efficiently, much faster, I think will actually be actually really important. I think the other challenge, and I think this is a historical component, is to do with bacteria. I think humans have had a different relationship with bacteria. If you look at sort of medics, so if you look at the history, we haven't known about bacteria for that long, only about 150 years, depending on when you want to count it. We were very slow, we being, the medical profession was slow into recognizing that infections were actually causal. And there's an education piece here because we typically think about bacteria as being causing infections. So sometimes people say, well, look, is the tumor infected then? And it's not infected in the same way that, say, somebody has pneumonia or a chest infection. This is something that I think is intrinsic to the way that cancer develops. But I think our relationship with bacteria also needs sort of redefining. So it's a really exciting area. I think a lot of things have come together, particularly from a computational point of view, from a data point of view, right now to be able to allow us to unlock that value and ultimately deliver that for patients.
Excellent, thank you. So basically, both technology development, data accessibility or availability, as well as an educational piece, which I really like that angle, as well as in just basically admitting there is this dysfunctional relationship between us and bacteria and their role plays, which I guess takes me to my second question, which is given that there is a lot of news or information or misinformation perhaps in the mainstream media or some news outlets out there that basically constantly describe the relationship between us and say the gut microbiome and the role it plays in regulating health, etc., etc. So that area seems to be maybe more advanced in the mainstream media, that there is talks about like the role the bacteria plays beyond the fact that it's just an infection. But that doesn't seem to be the case, say, in the medical space. Any thoughts on that and guidance for the audience in terms of how to seek better information when it comes to clinical sides of things?
Yeah, I think that's an excellent point. And I know there's a question in the chat that I'll answer in a second. Yeah, I think this is one of the challenges. There was a survey done and published in the BMJ, I believe, looking at where even the medical profession gets its information about the microbiome. And I think 80% of people get it from the mainstream media, which is, potentially useful in some regards because it highlights it. But obviously the scientific depth is obviously it isn't. It's at a very sort of certain sort of level. I think there is a lot of excitement about the microbiome from a gut perspective. I think the real challenge, though, it comes down to that maths problem, is that it requires a very different approach to it, because if you look at this from a pure association point of view, then you will always find one particular bacteria associates with a particular outcome. So, for example, my background is actually in sort of neuroscience, and we've seen this problem before. So when we scan people's brains and we do what's called functional imaging, we find areas in the brain that sort of light up, and they're associated with, some cognitive condition of X, Y, or Z. And there was a fantastic paper, which is probably about 15 years ago now, where they actually put a dead salmon within an MR scanner. And what they found was that there were biologically plausible activation within a dead salmon that actually related to some characteristics of the salmon. Now, this is a maths problem. It's not that the salmon wasn't dead, etc. It's a maths problem because you've got such a large search space. The conventional approach from a frequentix maths point of view simply is very challenging to be able to handle that large multidimensional data set. And I think that's one of the challenges with the general gut viewpoint as well.
Sorry, go on.
No, I was just going to answer the question in the chat about is it applicable to other cancer treatments like immunotherapy? Absolutely. I think the mechanism of action are very, very different. So we spoke briefly about checkpoint inhibitors, obviously used in a wide range of sort of cancers. There's a very large, has won sort of, Nobel Prizes, is a very large sort of blockbuster for certain pharmaceutical companies with revenues, in the 6, 7, 8 billion per year, just in the US alone. The papers have shown that actually, even though that's an intravenous drug, that actually the gut bacteria can actually influence response. So if you look at melanoma, typically a third of the patients would respond to a checkpoint inhibitor. What's been shown in a clinical setting with 2 back-to-back papers in Science, which are phenomenal studies, where it was the same patient, same treatment, same cancer, but they changed the bacteria, they were able to shift that by about a third. So a third more people responded, which is phenomenal. Now, the challenge is the way they did that was actually change their entire microbiome into somebody else's microbiome. Now, the difficulty is they don't know which bacterial group of bacteria or effect is actually driving that. And that's one of the key things that we're looking to actually sort out at BioCorteX.
Excellent. And thank you, G, for that excellent question. Again, please, if you have any more questions, feel free to post them. We will read them out. And so again, thank you, G, and thank you, Nick, for the answer. So just changing tack slightly from the regulatory point of view, say, for example, the FDA perspective, can you help us with thoughts on what it means for drug developers, knowing there is a new angle now to look at, which is can actually help in, say, selecting participants in a clinical trial?
Yeah, look, that's a great question, and I think there are different aspects from a regulatory point of view. First of all, there is no, as far as I'm aware of, quite rightly, the FDA and other regulatory bodies have extremely strict criteria about any sort of diagnostic or sort of screening tool that makes it into a patient population. And from a bacterial perspective, there hasn't been anything that's made it into that level, as far as I'm aware. Within the microbiome space, obviously, we know about H. pylori. We know about viruses and other forms of cancer. So, they have done, but not in this ecosystem type approach. I think what COVID demonstrated was that the regulatory bodies are actually recognizing that they can move much faster in this space, and I think it's something that's hugely exciting. I think it's really important though to as a clinician to recognize the limitations of what this work can do and the evidence base for it at the moment. I think it can really help guide early drug development, where we can look at trying to de-risk those pipelines. I think there's a lot more work that needs to be done before this can be moved into something like a diagnostic. But I think it's potentially really exciting.
Excellent. Thank you very much for that. I have one last question, and again, I'm asking difficult questions intentionally. So I guess looking into the future, and basically from the patient perspective, what do you think the future of treatment will look like for patients, knowing that bacteria play a role in response, potentially?
That's an unfair question.
It is indeed.
So whenever anybody asks me a question about predicting the future, there's always the sort of Keynes approach. If anybody into economic theory was one of the one of the great economists during the 1920s and 30s, and was very well known within the Cambridge sort of area. So when asked a question about predicting the future, his sort of standard response was that in the long term, the only thing that's guaranteed is that we're all dead. And I think that's the only. I think predicting the future is always challenging. I would like to hope that actually, bacteria and redefining our relationship with bacteria could lead to significant advances. I think it's one of the areas in medicine, one of the few areas in medicine I've ever seen where it's applicable across the range of scale. So from one individual to like large populations. I think it's one of the few areas of medicine where it's not a technique. We've co-evolved with these bacteria, so their ability to interact with our physiology is phenomenal. We have a bacteria in each of our cells. We just call it a mitochondria. I think there's a huge amount of potential, but I'm always nervous about trying to predict the future, so I'm not making gross predictions on this. But very exciting, I would say.
Excellent indeed, and wise. We have 6 minutes left, and it seems that we don't have any more questions. Nick, this has been absolutely phenomenal. Thank you very much. Madre, I can hand back over to you.
Oh, thank you very much, both Nick and Mo. And again, very happy Mo was able to get his microphone on so he could ask the questions. And for the very interesting questions that he asked. And thank you very much for the audience participation. Thank you very much, everybody, for joining us on the second webinar series. And we look forward to keeping the engagement going, where we next week will join and talk a little bit about regulation and market access. So I really would like to welcome everybody to hear a little bit. We're going to have Sharma Ragab and Dr. Ayman from BioCorteX who's going to be joining us. So it's going to be an interesting conversation. We're looking at from industry perspective, and we'll be inviting everybody to join us to hear how to enter the market in the GCC. And so please come with questions. And we just really want to thank again Nick and Mo for their invaluable contribution today. And please have a look out for the recording and share it with all your colleagues for everybody to get the information. Thank you, everybody, for a lovely day. Thank you.
Thank you. Thanks.
Have a lovely week.
Bye-bye.
Bye.